Here we go. So yeah. So again, where we left off yesterday is we looked at traits. We looked at those things called alleles, right? Things like eye color, hair color, of people. Those are types of alleles. For example, skin color is an allele. If you're looking at plants, maybe the size, the shape, the sweetness of a fruit, how big a fruit grows, whether the fruit has seeds or not. Those are all examples of alleles. They are traits. So we looked at what happens when we breed these things, and we eventually came up to something called dominant traits. And dominant traits override non-dominant ones, right? If you have on the top here the parents, if I breed two homozygous, the same alleles, two dominant, two recessive, I will get ones that are heterozygous. They look like they're yellow, but actually, if I breed these again, I can then get a whole bunch of green ones as well. And it's kind of just done through just basic like multiplication. We split up the alleles and we just draw a table with it. So that's simple and good when we have one trait. But what if I have more than one trait? Our biologist here looked at 7 traits, for example. How does he track those? Maybe he's looking at whether peas are yellow or green and whether they're big or small, for example. How can we map that information? So that's kind of where we're going today. So these things are called Punnett squares. And that's primarily what we're focused on today. And so what's eventually going to happen is next week, you guys are going to create your own like animals essentially, and you're going to breed them. We're going to do an experiment where you breed your own animals with other people's animals, and we see what we end up with. We're also going to eventually be able to work backwards. We're going to learn how we can work backwards up family histories to learn things as well. So again, we looked at things also called genotypes and phenotypes. Genotypes are the allele pairs. It's the genetics, genetics. The phenotypes is what it looks like. This P right here on the top right corner, it looks like it's a dominant one, but it has a gene, it carries the gene that it can pass on and make green peas if it breeds with the right pea. So now we're going to look at what happens if we breed 2 alleles. What if we breed 2 of these alleles? And this is going to be a little confusing at first. Once you break it down to a table, though, I promise you it's going to make a lot of sense. So we're still on 10.2. So we have a law here that's discovered, but basically when we have independent assortments, this means that when we have these alleles, these traits, these are randomly distributed during formation. And again, we will make sense of this, I promise. The key takeaway here is that basically this law means that all of these traits, these combinations are equally likely to happen. That's the real main thing we kind of need from this. So again, what we really just kind of need to know about this is that what I've done here, and I'm actually going to, I'll enlarge this a little bit. So what I've done here is I have enlarged. Sorry, are you still writing?
No.
Okay. So this is actually, I'm breeding 2 things here. I'm breeding one with a dominant and recessive gene, and then the other one, let's say this is, I don't know, color, and then I have another one of whether it's round and recessive. So I have a cell that's like this. It's a parent cell. And then as those gametes get formed, these combinations are all equally likely. Because I'm only passing on 2 of my genes. I pass on half of my DNA. So how we do this is we use those Punnett squares. This is what we did. You do not need to draw this. You don't need to draw this because you've already done it. It's this thing. This is a Punnett square. So it's a nice, helpful way to be able to try and predict what's going to happen in a generation. It's especially helpful when we're passing on two traits. So again, we looked at like these Punnett squares with yellow or green peas the other day. So where this gets a little bit weird is, again, what if I am breeding two things, a parent generation with two traits, yellow or green, round or wrinkled, two alleles. I have a boatload of combinations here, right? But there's still some things that I've kept the same. You can see here, I have 4 green peas and I have 12 yellow peas. It's still a 3-to-1 ratio with that, for example. You can also see that I have 1, 2, 3, and 4. I know that's a little bit harder to see, but I have 4 wrinkled peas and 12 round peas. So that 3-to-1 ratio is still going to stay consistent. You will know if you've messed something up if the trait that you're trying to reproduce does not have a 3-to-1 ratio. Something went wrong somewhere. So 4 different types of alleles from the male's gametes, and 4 types come from the female gametes. So it's the same thing. I've just added on an extra gamete. And what I get is I get a new ratio here of 9 to 3 to 3 to 1. There's, like I said, there's still that 3-to-1 ratio with certain things, but now I have four possible options. I could have green round peas. I could have green wrinkled peas. I could have a yellow round pea or a yellow wrinkled pea. So I have 4 different options there now. Go. Again, I've just enlarged that picture here. So here's what we're gonna do. I'm gonna give you guys these things here, these traits. So the Y, let's denote these. So the Y in this case, wait, what am I doing? I can literally type it. So you guys are gonna get a chance to actually practice this out. Let's try it out. Here we go. So we are going to breed these two things, text. Here we go. So we're going to say that big Y's, I'll make that bigger here. We're going to say a big Y means that it is a yellow. We're going to say small y is green. We're going to say R, we now have R's, are the roundness of it. I know it's kind of hard to see, but this is wrinkled here. So whether it's smooth or it's wrinkled. And a small r is wrinkled, it's bumpy. So I have 2 different alleles, 2 traits that can be exhibited 2 ways. So there's 4 possible things here, right? So what we're going to do is we're going to breed these and we're going to try and predict what the heck happens. So first off, in our first generation, we were only going to get which one do I want to breed actually? Yeah. So I'm going to get you guys do is I'm going to get you guys to quickly draw a 4x4 grid in your book here. And I want you just to make sure you guys put this information in there. So I'll give you a second just to kind of copy that down. Let me make this smaller. So what we're going to do is we're going to split this stuff up. If you guys remember this thing here, it's saying that these the dominant gene is going to go 2 ways. The recessive one going to go 2 ways. The dominant round going to go 2 ways. The recessive round going to go 2 ways. Basically, what's going to happen is we're just going to slowly kind of piece this together here. So if I'm thinking about this top left P, I'm just going to go through this first one I'm crossing out here, the Y. So I'm just going to put just the big Y first, the dominant Y. on two of these. And then my other gene that I can use is this other Y. In this case, it's pretty simple because both are dominant. So you know that no matter what, the dominant one's getting passed along by the one parent. But now I need to, so I've contributed my half of DNA for the color. Now I need to contribute my half of DNA for whether it's round or not. So I can see here that what I have is I have two dominant round genes as well. So no matter what, it's going to be two dominant genes passed on. And if I've done that, I've completed my first parent. What I now need to do is do my second parent. So again, I'm going to do the exact same thing here, right? I'm going to look at the first gene I can pass on. The first one I see is a green gene, lowercase y. I'm going to pass that on to both these options here. Because this lowercase y can either go with the first R or the second R, both of which are recessive. And so what I eventually get is just this across all four. So you can probably predict what this is going to look like in each of these cells now. All I have to do now is combine it. Combine the Y's, combine the R's. And I do that for each cell. Now you've probably started to notice the pattern that's going to emerge here. Every single one of these is going to be the exact same. Specifically, they are all going to be a smooth yellow pea. When you breed two homozygous things, you don't get a lot of genetic diversity. So what we've done is we've breeded the parents, and now we've gone to the first generation. So what we're going to do again is now we're going to breed these offspring. I can pick any one of these peas, and now we're going to get some incredible genetic diversity. So what we're going to do is we're going to repeat the entire cycle all over again. So I'll give you a second just to make sure you got that all written down. Okay, so what we're gonna do is we're going to now breed the first generation, the F1. So sorry, you're gonna do one more Punnett square. Just one more, I promise. So this is the one we're breeding, YYRR. In this case, if you're a little confident, try and put the allele combinations here, if you're able to. Because you're eventually gonna have to do this yourself. So try and put those in there if you're able to. If you're not sure, it's fine to guess, write it in pencil. I'll give you a minute to set that up. Okay, so we're gonna start filling this stuff out here. So again, if you're unsure how to choose, like which alleles do I pick here? How do I know what's gonna end up here? Well, I'm gonna look at my first cell here. I'm gonna take a dominant gene for color, big Y. And that's gonna go to two different locations, right? Again, if I'm drawing that, I will erase this in a second, but it's gonna go like that. And then the other one, the recessive one, is gonna go in two spots as well. There's 4 combinations. I'm just gonna erase those lines. So next, I'm going to take my pen. I need my pen tool. So I've done that. I've crossed that out. I've done both of those. Next, I'm going to take the gene for whether it's round or not. I'm going to start with the capital R, the dominant one. Oh, sorry. And that can get past 2 different spots. And then I take my lowercase r and I put that 2 separate spots as well. And so because of these are the exact same cell, I don't need to repeat the process with the other one. I can literally just use the same ones because I'm breeding the same thing. They're going to have the exact same combinations. But so now what's going to happen is as I start to breed these and show the combinations, we're going to get some pretty tremendous genetic diversity. The first thing I'm going to get is I'm going to get double dominance in both, right? But as I work my way across, double dominant in color, but now I carry a recessive gene for whether it's round or not. Heterozygous in color, one dominant, one recessive, but homozygous, double dominance in roundness, and so on and so forth. So eventually, I'm going to end up with this. I want to actually copy those. So this, what we have here, right now, am I displaying the genotypes or the phenotypes? So what I have on the screen, are these the genotypes or the phenotypes of these peas? These are the genotypes, right? These are the genetics of it. It's not what the peas actually look like. What the peas actually look like, though, is this. So these are just the peas for, right? The second I have a dominant color, it has to be yellow. But I do have a couple ones that are recessive. The second I have a dominant R, it has to be smooth. And this is an incredibly pixelated image, so that's hard to see. But I do have 4 round ones here. Oh, sorry, we have 4 wrinkled ones here. And so we still have that 9 to 3 to 3 to 1 ratio. What's the 1? It is this little p down here, right? It is homozygous recessive for both. So I get, out of 16, only 1 of them get that. 3 of them are going to be green, but have a dominant round shape. 3 of them are going to be yellow, but are going to have a round shape. And then the other ones possess both dominant phenotypes, not genotypes, phenotypes. So that 9 to 3 to 3 to 1 ratio is what it looks like, not its actual DNA. And you could go one step further now, right? So like this is where genetic diversity comes on. What now happens if I pick, this one that I'm boxing and I breed it with any other one? What if I breed it with this one, right? You'll get completely new genetic diversity that arises from this. This is eventually, what leads to the theories of evolution. This is how we breed dogs. This is how we breed plants. This is how we help feed the world. It all comes down to Mendelian genetics. It's all come down to this simple pea experiment. We've advanced it tremendously, of course, but that's where it all starts. Okay. Are we done with this? I feel like we might be. Yeah, I think we are. Okay, so that's Mendelian genetics, and that's Plunkett squares. And you are going to eventually create your own Plunkett square for something. That is coming. But let's put a little bow on Mendel for now. So we've done that already. This is the actual data here. Now, one thing to note is his stuff was not exactly this ratio because it's still a little bit like random chance, but in theory, with a large enough number, if he were to do 1 million peas, this would get really, really close to it. Here's his actual numbers, right? These are his actual numbers here. that he did. So the actual data might not match the predicted ratios. Okay, so I got just 2 questions here for you. Just 2 questions. Fair amount of space though. You're going to want probably 3 lines. And I'll get to number 2 in a second. So I'll give you guys just a minute. So we're starting out. What does a Plunkett square do? We've been using it for 2 days. Plunkett square shows the combinations of alleles, of genes. And we can use it to predict the phenotype and genotype, basically. So it lets us breed things and look at the outcomes. This is a very fancy way of saying it lets us breed things and look at what's going to happen. So next, what we're going to do is we're going to make a Plunkett square with, what if we have fruit flies? So fruit flies are something that are used extensively, actually, in medical research. You can look at how well are they going to be able to live, how long can they live, how long can they die if you affect different things with their diet or with their environment. So the reason they're useful is because they breed like crazy and they have an incredibly short life. So you're able to go through generation after generation after generation very quickly, cheaply, and easily. So what happens if I have, right now fruit flies, with the red eyes, it's dominant to pink eyes. We want to look at, first off, what's going to be the phenotypic ratio of a cross between a heterozygous male and a pink-eyed female. It's a very scientific sounding question, right? It's a very scientific question. But all we really need to know here is, I'm breeding a heterozygous male and a pink-eyed female. Let's see what happens. So if that's the case, I'm going to end up with something like this. It's a heterozygous, which means these are different for the male. But I know it has red eyes, so it needs to have at least one dominant gene. Whereas the female only has pink eyes, which means both of those have to be recessive, right? Because if it had a dominant gene, it'd be red eye. So I'll give you a second to fill that out to the best of your ability. Again, I just combine these things, this is what I get. But the question was not complete a Plunkett square. The question is asking, what is the phenotypic ratio of breeding these two? What does a phenotypic ratio mean? Well, a phenotype is, what does it look like? And in this case, it can only look like two things. It can have red eyes or pink eyes. How many of these have red eyes of the generation, of the next generation? 2. It's going to be a 2-to-2 ratio. Or if you round down, that's a 1-to-1 ratio. Half of them will be red-eyed, half of them will be pink-eyed.